Our Next Generation Leader program is a program that's now, I guess, just starting its third year. And the Next Generation Leaders were selected as people who are outstanding in their field, early career scientists working on topics and questions that were related to the kind of questions that we're interested in here at the Allen Institute, but also had great achievements in their own field already in the short careers that they'd embarked upon already. So our first batch, or our first class of Next Generation Leaders is here with us today to give a little of their input and a little bit of their perspective, both on their opinions of the sort of state of field of neuroscience as it is now, as well as their own experience as Next Generation Leaders. So the format of this is going to go where they're just going to go through some introductions, each of them, for a couple of minutes at the beginning. And then we have a set of panel questions that they'll discuss amongst themselves for about 15 minutes or so. And then we're going to open it up to questions from the audience for the final 15 minutes. So please join me, first of all, in welcoming our Next Generation Leaders. Okay, so Yuli, why don't we just start with you?
Okay, so this working? So hello, everybody. By now, most of you have probably spoken to me in some fashion. As Amy told you, I'm part of the first class, which started in 2014. So I'm just going to give you a brief overview of what I do and where I come from, so that those of you who don't know me have some background. So I'm at Cedars-Sinai in Caltech in Los Angeles, where I started my own lab in 2012. And I focus on working with human brains, and I do in vivo recordings. So my specialty is to do single neuron recordings in a variety of neurosurgical situations where this invasive brain access is required for clinical reasons, and then we take advantage of it and do basic research by inserting additional electrodes which allow us to record single neurons, which is for the most part microwire electrodes. And using this technique, we study very basic questions, and my main interest is single trial learning. So I showed the patients sequences of images, and the first time they see the image, it's novel. The second time they see it, it's familiar. And that kind of learning, which forms episodic memories, is something which is very difficult to study in animals, to have to train an animal to do this. So we have made quite substantial progress in understanding what happens, what's different between when you see something the first versus the second time, and how is that translated into a decision, and how can you rate that decision by asking for a confidence rating. That's my principal interest that I have spent some years working on. So my relationship to the Allen Institute has been mostly through the effort that you have made in the human brain. So I think that's very unique and relates greatly to that kind of work, and it contributes very unique information that I think nobody else could have done at that scale and at that quality. So I think it's for my particular field of neuroscience, that is going to be the most influential aspect of the Allen Institute.
Thank you.
Hello, my name is Chen. I'm a new assistant professor at Boston University in the biology department. I just started this year. My lab was finished renovating 2 weeks ago, so we're just moving in. Before that, I was a postdoc with Fritjof Helmchen in the University of Zurich, where I was there for about 5 years. And the lab that I'm starting up right now is interested in studying how different parts of the cortex interact with each other. What are the rules that govern long-range cortical communication? And I do this work in the mouse whisker system. So we're looking at interactions between the barrel field, primary somatosensory cortex with secondary somatosensory cortex, as well as primary motor cortex. And we're using a lot of optical approaches. We do some technology development where we've developed microscope 2-photon systems where we're imaging now across cortical areas simultaneously, and we can combine these types of functional recordings with an anatomical tracer, so we can label specific feed-forward or feedback neurons between these areas and start to look at specific dynamics across these connected neurons. And these are the kind of areas that I'll be interested in pursuing in the years to come. And so my research very well aligns with the work that's being done here with the MindScope project and the Brain TV project. So it's been great to kind of track, see how things have evolved here over the past 3 years.
So hi, my name is Nevel Sanjana. I just started my lab earlier this year at the New York Genome Center and also in the Department of Biology at NYU. And before that, I was a postdoc with Feng Zhang at the Broad Institute in MIT. And so over the last few years and continuing on in my lab now, we've been working on new genome engineering technologies really to enable us to do genome engineering in non-traditional genetic model organism systems like human cells, and to really to do genome engineering at scale to test out thousands of different hypotheses about genes that might be involved in different health and disease states. And so a lot of the applications for these things like genome-wide pooled CRISPR screens, where we're knocking out every gene in the genome, have been mostly in areas of cancer, but also some work with neural development and de novo forms of autism. So some of what we're interested in doing is taking a lot of the sequencing data that's coming out of sequencing patient populations and being able to test, again, at scale, a lot of these different genetic interactors that are thought to be associated with the disease to see if we can really see, does this particular mutation cause a disease? And so in the last few years, we've been taking advantage of the fact that this is a DNA level technology, that we really are editing the DNA itself, and instead of just looking at that 1 or 2% of the genome that codes for protein coding genes, we can now look at the 98% of the human genome that's non-coding and really try and find what are the different knobs and dials that affect gene regulation. So we've been doing things like taking large non-coding regions and doing saturation mutagenesis screens to find what are the functional elements here and how do they correlate with other marks of function in the non-coding genome, like epigenetics and things like that. My interaction with the Allen Institute has been fantastic. I've been very impressed with so many of the programs here. I'm also it was very nice to see yesterday this introduction to the Cell Science Institute and see a lot of great, very careful genome engineering work happening there. So I've just been very happy to be a part of this.
Hello, I'm Chris Saha. I'm an assistant professor of biomedical engineering at the University of Wisconsin-Madison. And my lab is very interested in cell and tissue engineering from pluripotent stem cells, as well as other accessible human cell types, as well as things like T cells and things that we can engineer pretty readily from routine clinical procedures. And our lab is really focused right now. Half of it is on kind of genomic engineering, correcting mutations, putting in reporters, as well as putting in a suite of emerging synthetic biology gene circuits into human cells. Right now, mostly at the stem cell stage, but then we take those engineered stem cells and then make neural tissues from them. It's being used for a host of various applications, disease modeling. Specifically, we collaborate with folks that look at inherited forms of neural developmental disorders, Fragile X, Rett syndrome, some actual inherited retinal disorders as well. And the other half of the lab is really interested in kind of targeted epigenomic engineering, so cell reprogramming, really transitioning cells from skin states to neurons, as well as to pluripotent stem cells. I definitely want to thank the Institute for having this type of council. I think personally it's been fantastic. I've learned a lot about really a new type of way of doing science and how it interacts with really the larger community. And it's really been fantastic to see where the field is and where it's really thinking of going.
Hi, I'm Lisa Giacomo. I'm an assistant professor at Stanford University in the Department of Neurobiology. I've had my own lab for almost 4 years now. My lab tries to understand our brain's sense of space. So we now know that there are neurons in the brain that allow you to have all of the components that you would really need to generate a map of space. There are neurons that act as a compass, a longitude and latitude coordinate system, or tell you where boundaries and places are in the environment. My lab really aims to understand the mechanisms that generate these different spatial neurons and understand how these neurons go on to drive behavior. I think in terms of the Allen Institute, we've been very inspired by the work that's going on here. I think my field is acutely aware of these questions regarding how we best can define a cell type and how we can extract more data from more cells. And I think these types of approaches really have the ability or the potential to revolutionize how we think about processing, not just in primary cortical regions like visual cortex, but these deeper non-sensory cortical regions that my lab is very interested in.
Great. Well, thank you, everybody. I had a few questions that have come sort of from general questions that a lot of us here at the Allen Institute have, just to reflect on some of your experiences, both as outstanding young investigators, but also through your role as next generation leaders here. So one of the questions that we had was related to your challenges that you've experienced as young investigators. So all of you have had already accomplished careers, but like everybody else, there have got to be some bumps along the way, and we're just curious about some of your perspective, specifically on a particular challenge that you've had at your early stage in your career. So Chris, I think that you had some thoughts on this.
I can start. I think there's kind of some familiar answers, funding, recruiting great students. But for me, I think the challenge that I thought about most deeply and took the longest time for me to kind of come up with a strong answer is fit. Exactly how your work and your perspective fits in with the other work being done really in the field. So a lot of us have to, I think, differentiate ourselves from our postdoctoral advisor, as well as other PIs that may be, funding, have very large pots of funds and heading big institutes. And as you're starting out a lab, you have to be realistic that you will only have one postdoc or grad student at the beginning, and you also will be the other person in the lab doing things. So, I think all of you kind of go through that process as a postdoc, understanding where your perspective is really unique. I mean, you argue that in your papers, you develop that in your kind of interview process, but I think it still continues as you start doing your first experiments. I think all of us will say that we didn't actually do the experiments we pitched at in our job talks. I mean, not exactly to the T, things change. For me, I interviewed in 2012. I talked about gene editing with talons, and CRISPRs came along 6 months later, and in certain ways I had to kind of figure out how I fit in that field, as it was really coming on really quickly. The other, I think, component of fit to me that was interesting was kind of really knowing deeply what the institution thinks your role is. So I think when you're hired for a reason. You're filling a gap, I think, or bringing some sort of expertise that's complementary to what's already there. And I think it's really important to know deeply what that is, because there's all sorts of larger projects that I think that, for me, the department and the university, as well as kind of multi-university centers, are trying to do. For me, it was something like cell manufacturing with human cells, or maps of cell types in stem cell derived tissues. These types of kind of really large efforts, and all of you here probably are familiar with very large efforts. But at that, really figuring out what you would bring to that process is, I think, important as you contribute to that, but then also have really something that's your own real new question. So to me, those types of questions took me a long time and were challenging, because people will have their own answers, but it is really your next big step and what you'll be spending 60 to 80 plus hours a week trying to figure out.
So that sort of segues nicely with one of the next questions, which was related to what's possible now that wasn't possible when you started your lab. These things do change and modify their direction. And Yuli, I thought it would be a good thing for you to address because the field's moved, as you said, sort of in your introduction, quite quickly in terms of some of what's possible with human brain research. And when you first started out, things were in a slightly different space, even just in the short few years.
Yes, absolutely. So, I mean, that question is very difficult to answer. There's obviously many, many topics that I could cover, so I'm just going to mention a few things. For me personally, there's a few things that stand out for what I particularly do, and one of which is the advances in electrophysiology. And that hasn't really penetrated yet into humans, but it has to some degree. So what I mean by these advances is that now suddenly it's becoming fairly routine, or at least imaginable, that you could record 1,000 neurons at the same time in a sort of fairly standard way. It wouldn't be outrageous anymore to claim that. So that really for me has been one of the highlights of the last few years, that technical problem seems to have been solved, and not to a fairly large degree due to the work performed here. And this has penetrated to some degree into humans. There is now some new technology where it has been shown that one can make very flexible arrays which are very thin, and they're plated on polymers which can be put on top of the brain under the dura, but which don't penetrate, with active electronics embedded, much like the IMEC probe, which allows recordings of very finely restricted electrical signals, which in some cases even allows to resolve single units, which are without penetrating the brain. And yes, they're superficial neurons, and it's not going to go very deep. But that kind of technology where active electronics is embedded in a soft material which is embedded in the brain, to me is a really fundamental advance that has come along. And this also includes these polymer electrodes, which are very soft, where the actual recording pads are plated not on silicon, but they're actually plated on a soft material, which is then inserted into the brain, essentially glued on top of a small wire, and that glue dissolves when it comes into contact with ACSF. These kind of technologies, to me, is really a fundamental advance. And the second thing that to me is really striking how far it has come is the concept of a cell type. I mean, to me, if you had asked me 4 years ago how many functional cell types there are in V1 or in the hippocampus, the number I would have given you would probably be very small compared to the number I would give you now. And this concept and how refined we can think about it, and we can define cell types electrophysiologically or morphologically and genetically and mixtures of that kind of thinking, to me, is really novel. And together, these advances really reinforce the point that now the problems are not at the data gathering stage anymore. We have pretty good ability to record data. We can define cell types in various ways, including in humans. That's another thing which wasn't imaginable to me even a few years ago. So now we can really actually start thinking, what does this mean? We recorded 1,000 neurons, which are distributed among 100 cell types in a behaving animal, perhaps a human. Now, what are we actually going to learn from this? That seems to be the frontier now to me. It's really moved from, well, we can't really record, so we don't know. Now, yes, we can record, so we do know what these neurons do, but now what does it actually mean? So to me, that's the frontier now.
So Lisa is doing something somewhat related, I think, with those kinds of highlights that resonate also with the research that you do, but coming from a very different direction. I was wondering if you could also comment on the technologies that are really creating an impact in your work and in the field from your perspective.
Yeah, so I would echo a lot of what was just said, but also we have a slightly different perspective because we work in these non-sensory cortical regions where it's not exactly known what these cells are really representing. One of our challenges has been that or I guess one of the exciting things has really been that there is a sort of general consensus now in neuroscience that there is a lot of data in populations of neurons that just really can't be captured when you only look at the single cell level. So there could be features of the environment or features of behavior that can really only be observed when you consider these very large populations of cells. And I think to see these technologies that now are potentially going to allow us to do that, both the electrophysiology and also this wide field imaging, that really opens up the technology for us to sort of start approaching questions that way. And I think we're very excited about these very large-scale electrophysiological recordings because a lot of the brain regions that we work in are very deep structures. And this is a better way, a good way to access those regions. We also are interested in looking at the dynamics between large populations of cells across cortical structures, deep cortical structures. So that's another feature that we think is really fantastic. And I also think that there is population data that's important not only in how individual cells are representing information at any given time, but the relationship between those representations. And so I think all of these technology developments that are kind of geared towards getting access to more cells at more cells at any given time is really going to revolutionize the way we think about how the brain is doing or is supporting behaviors like navigation and memory. And I just would maybe add one other thing in terms of this cell type question. I think it's been very inspiring to see how the Allen Institute has really addressed this idea of what is a cell type. In our own lab, we've sort of struggled with the same thing. We typically, or sort of historically in our field, define cell types based on their function or their sort of tuning curve. So what sensory stimulus elicits the largest change in a firing rate. And it turns out that it's really hard to capture what cells are actually encoding when we just use that sort of single way of defining what a cell type is. And so we've started to move towards using more machine learning or statistical approaches to try and, in a very unbiased way, define what variables does a cell actually care about. On the other hand, my lab is also very interested in trying to identify cell types based on sort of molecular features or molecular characteristics or of the cell, and trying to decide how to actually wed these two ideas or how these two ideas might sort of come together, I think is a very deep philosophical question that my lab is not only struggling with, but the Institute and I think neuroscience in general is sort of struggling to come to terms with. So it's been very inspiring to see how a large group of really talented scientists that have this wonderful collaborative kind of team approach have tried to address this very sort of deep question of what is a cell type and how do we actually define a cell type on these different sort of scales.
Yeah, I think that resonates with a lot of us here as well. And thinking about both the tools from the technology perspective and the timeliness of some of these questions, I was interested in your perspective specifically on, gazing into the future. So over the next few years, 3 to 5 years, Nevel, maybe you can share with us a little bit about where you think things are going, or with some of the tools in hand, as well as the trajectory of development of ideas and maturity of the field. What are you seeing in the future for us?
Yeah, so since I'm a little, I feel like my lab is split between some neuroscience projects and some genetics projects. So I'll interpret the question maybe broadly. And I think what's clear just in the day and a half that I've been here in this visit is that with so much data, it's kind of, people always talk about big data, but I feel like this place is really like rising to the challenge in terms of building the software tools and the infrastructure to really handle it. And I think academic labs more broadly will need this and will start to depend on this, because otherwise, when you collect a lot of data, it's just not useful if it's not in an accessible, nice format to play with. And genomics is doing, I think, a decent job with this, but neuroscience, I think, is largely, it's a very difficult problem for neuroscience, but it's being handled really nicely here. And just some of the things that I think are interesting from my perspective is I think there's this huge effort on international effort now looking at cell types. The NIH and I think Wellcome Trust and folks have funded this large cell type atlas. And so I think the Allen Institute should be very proud of kind of being ahead of the curve with this. And I think you're going to see it now, not just in neural cell types, but maybe cell types more generally, with at least transcriptomic analyses. And I think a parallel kind of effort in my mind is not just in the same sense that we want this comprehensive cell type atlas, something else that's missing that our lab is working hard to address is to really understand what are all the functional areas in the genome itself. And so I think in the post-genome era, since whatever 2001, we've done a decent job of learning about what different genes do. But as I mentioned in my little introduction, is that the genes are just an astonishingly small fraction of the 3 billion bases of the human genome. And that really it's becoming clearer and clearer that there's many, many functional elements that are embedded in non-coding regions. And I was very happy to see that this is my first visit here in about a year and a half, that a lot of people are talking about enhancers, both in human tissue and mouse tissue, and doing things beyond RNA sequencing, beyond counting gene expression, like ATAC-seq and other assays for these enhancers. And so I think this is, for the field of genomics and genetics in general, this is a large future project, is to really unearth all these functional elements. And in the non-coding genome, there's no central dogma of molecular biology. We don't know, you don't have this nice DNA, RNA, protein thing. So I think it's a much more difficult task to say what is a functional element in the non-coding genome. And I think that's going to be important, of course, for cell biology broadly, but also for neuroscience, because there is so much recent evolution with brain function. And it's likely that it's in these non-coding regions, which are very poorly conserved with mouse. So I think using human, a lot of people have talked about human model systems here, and that's so unique to this place. So I think doing some of these kinds of non-coding manipulations or understanding non-coding function in human models is going to be an amazing future direction to move in. Yeah.
Thanks. So on a sort of closing note, first, just some of the discussion here. We've certainly at the Allen Institute benefited greatly from your experience as leaders in your field. And I thought it would be really nice maybe to hear from one or more of you about how your experience as a next generation leader for us or with us has shaped or changed your perspective or your science. And Jerry, I was wondering if you could share maybe a little of your perspective on that.
Yeah, I think I have maybe 2 points to offer. One was just to be exposed to a model in which science is being done here, which is quite a unique model compared to academia in terms of a lot of teamwork and kind of trying to do science on a large scale, and how do you actually execute these types of kind of projects that in terms of the science that you guys are doing, and also the same kind of questions that I'm interested in. And I think there's trying to think about in terms of when I'm starting the lab, and the lab is going to be much smaller than the institute I have here, but I think there's going to be kind of modes, things that I can adopt in terms of kind of pipelines and how to kind of standardize, automate certain ways to kind of help kind of further along some of these kind of questions that I'm interested in pursuing in the lab. The other thing that was quite tangible in terms of my experience with the Next Generation Leader program was that during my transition from a postdoc to PI, I had a brief amount of time where I was able to come and to the Institute and for a couple of weeks actually do a little bit of work. So I saw there was a lot of interesting work being done, a lot of techniques that I thought that I could use and employ in my lab. And so I came and collaborated with a few people here, learned a few techniques, got a little bit of data that I was actually able to put into a grant and actually get funding for. So that was something very tangible in terms of my interactions here, and something I would suggest the kind of first and second year next generation leaders explore.
Well, that's great. Thank you, all of you. We have about 10 or 15 minutes for questions, plenty of time for the audience to pitch your questions to our most senior next generation leaders here. Even though they're maybe at the more junior stages of the career, they've already accomplished a lot and would have a lot of, I think, positive advice for members of the audience. So were there any questions that people had for any of the panel members here? At the very top, I see.
Okay, this is for the man in the orange shirt.
I think that's
You mentioned genes and the correlation between the body and the brain and the body, kinda? Is that
Kind of,
it?
yeah.
Okay, you know, you've seen me in this walking brace, and my doctor at Group Health said this walking brace is sending signals up my spinal cord to my brain saying, this is the way you walk, this is the way you walk. So, how prevalent is your research to basically my question is how can you guys transfer your information over to the medical field and work on bettering outcomes of rehabilitation and functionality?
Yeah, so I think that's a great question, first of all. I think there's a large spectrum kind of biomedical research, and actually the work we do is we try to be very close to the translational side of things. So we're very interested in what are the particular, for me as a geneticist, what are the particular genes that are involved in a disease? What are the particular ways that if a cancer is resistant to a drug, how can we understand what are the genetic mechanisms of that drug resistance? Because that dramatically impacts that person's life, right? If that cancer is drug resistant, metastasizes to a different part of the body, that's a very serious thing that happens. And so we try and, in my lab, develop tools that are for manipulating the human genome with the very direct purpose of understanding those genetic interactions. And so we don't do so much, I think, on the neural regeneration side. Maybe Chris would be a better person to talk to about that. But at least in terms of the spectrum of where we are in the research side, we really are try to stay very focused on what is the most immediate impact these new, very basic discoveries can have on the translational side. Maybe stem cell biology. I don't know, Chris.
So I work with a team of regenerative medicine clinicians, and they're interested in potentially putting biomaterials and/or stem cells together, or stem cell derived cells into injured or diseased areas, and it's usually a very long process and is risky, as many investors would probably tell you. But I think there's probably lots of different researchers in that space, both across academia and industry, that are, I think, talking to all of the kind of basic scientists, people that are more hybrid like me, that are also kind of closer to the translational side, thinking hard about what some of this new knowledge is doing and how it could go into making those strategies more robust and effective.
Yeah, I mean, when we spent a lot of time talking about cell types up here, and I think that might just sound very abstract. Okay, there's a lot of cell types in the brain, but I think if you wanted to make a particular type of cell for some kind of regenerative purpose, you have to at least know where you want to go first, right? So if you want to screen a bunch of transcription factors to say, how can I make a dopaminergic neuron to read, transplant into a patient, we first need a really nice characterization of what is a dopaminergic neuron in terms of its maybe full gene expression profile. And so I think these are all baby steps toward that kind of goal.
Yeah, I mean, just to add on that, I mean, I think one of the one of the really great events that I've been fortunate to be part of was a scientific advisory board meeting here at Allen, where I met Lawrence Studer, who's exactly thinking about making cell types for implantation into the brain. And he says he struggled for many years and a long portion of his career to figure out exactly what cell type he should make from a stem cell, such that it would be the most efficacious cell. And that's one model of doing that type of research. And I know it's not the only model. And I'm also part of a National Academies committee that's really thinking hard exactly how do you define the cell type that will be transplanted? What are the standards? How do we make sure that the clinic that's offering it, perhaps in Mexico or abroad, is really doing it in a very stringent way? So developing standards built on some of the knowledge that's being generated here and elsewhere are super important to try to make those types of, I think, translational practices more safe and effective.
Well, I'm with the Brain Injury Alliance of Washington. I'm on the board of directors. And if we can help you guys in any way, I've got our card to give you guys, and we'd be more than happy to help you.
Thank you.
Thank you for the question. I think a lot of us here really immerse ourselves in some of the basic research, but ultimately the goal is to apply that research and at many different levels. The basic research, understanding individual cells and the components involved, whether it's genes or whether it's the functions of the individual response properties of the cells. Ultimately, this is an interest to all of us. So I'm wondering if there are other questions that people have for our panelists. I think there's
Preferably easier questions.
But that's one of the most important
It's an
ones,
important one, yeah.
honestly.
All right. Well, I hope this is easier. But what of the existing products from the Allen Institute do you find most useful in your research?
Well, I can perhaps start off, and that the detailed anatomical atlases of the human brain are certainly extremely useful for the type of work I do. It's just, if you look in the human neuroanatomical literature before the human brains of the Allen Atlas became available, they are literally based on a few people, like the My Atlas, which everybody uses. So now the kind of detail that the Human Brain Atlas offers, especially the latest one, I think is phenomenal and personally very useful to me.
Yeah, we actually also, we really leverage a lot of the online resources. My lab's very interested in understanding how specific ion channels contribute to coding across multiple different brain regions. And we've had a number of lab meetings that have just started with tools and information that we've gained from the Allen Institute websites in terms of where our ion channels are expressed, what that looks like in a developing rodent versus an adult rodent. So those online resources we certainly use. In addition, we would be extremely interested in testing out technologies like the NeuroPixel when it becomes more widely available. We're always looking for new electrode arrays to let us allow us to gain access to more cell types, and we've been very impressed with the progress on that particular product. So that's something we would love to integrate into the lab when the time comes.
I have a question up here. Hello.
Yes,
Hi.
Alan.
Hey. So I've heard the themes of things that you guys have learned from the Allen Institute around team science, some of the industrial scale approaches we're taking, but what about the open science? So one of the things that I often hear is pushback, and certainly from you guys, you're young investigators within an academic setting. It's really hard. We have to protect our data. We have to get published in order to succeed in our careers. I've always been hopeful that a new generation of scientists are coming along, such as yourselves, who want to change that paradigm. So I'd love to hear your perspective on changing that paradigm.
I think that it is changing. I think that if you look around, and people are, actually publishing their manuscripts before on bioRxiv, some of these online sources before their papers have been officially accepted into a journal, and these kind of manuscripts are being cited, and even at that stage. So I think, and these are kind of been done by a lot of these younger investigators. So people are more interested in kind of putting out their data, before it's kind of officially ready to go, and also there's more avenues for kind of hosting or providing your data sets kind of in an online format for people to kind of use and do further analysis with and so forth. So I think it is kind of changing slowly, and I think we're all very enthusiastic about making science in general more open than it has been in the past.
I would agree with that. I think it is changing. I think one of the challenges, though, in terms of being a young investigator is that it can take us 3 or 4 or even 5 years to collect a data set. And once we have that data set, we tend to like to get several publications from that data set. I think things that could really be useful in terms of promoting this type of open source data sets in terms of young investigators or sort of individual PIs is funding schemes that really promote collaborative and open sharing of the data. If I know that that's a component of how to get funding, then I'm certainly more motivated to make sure that that is implemented well in the lab. And I also think that from the higher up group, the more senior scientists, I think there needs to be acknowledgement within universities that collaborative science is just as meaningful as individual contributions. So in terms of just the tenure process and the promotion process, that there's acknowledgement that using an open source data set or providing your data on the open market is as significant of a contribution as holding onto your data set, and trying to eke as much out of it as possible. So I think those things will continue to foster that. But I do think that there's a change towards a general kind of mindset change, that this is an important thing that scientists should be trying to do.
Yeah, I mean, to add on that, NSF and NIH have pretty strict requirements now about data management. It doesn't say sharing, but I think most of the younger generation interprets that as sharing in some form or another. So certainly the challenge that Lisa talked about are still there. But I think kind of more generally, apart from data, other materials and reagents are being shared to me at pretty much higher, almost a higher order of magnitude than before when I was doing my postdoc. One example is AdGene.
It's been a game changer, AdGene,
yeah. In the CRISPR world is really phenomenal how often and how quickly plasmids move from lab to lab. Materials, cell lines, I think the Cell Science Institute, is putting out things through Coriell. Certainly there's kind of I think a total change in practice at Wisconsin of trying to get things banked and distributed really broadly and openly. So yeah, to me, I think the change is already kind of apparent in many ways, and it's going to even further change.
Yeah, also with analysis and computational modeling, now with ModelDB, that's definitely become more open source as well.
Yeah, in the field of human recordings, I think the message of open science from the Allen Institute hasn't quite taken root yet, but I certainly I think the success of the open science model that you have demonstrated is really a good example to show people that this can work, because the reality is, and you know this from your own data sets, you look at maybe 5% of the data, and you only look at what you're interested in, and the rest nobody ever looks at. And for data which is extremely hard to acquire, that's unacceptable. Somebody else should be able to look at that. So from one particular experience I recently had, which is that if you get a grant from NIMH, they will actually require you to upload your data, the raw data. So I don't know if they have taken a cue from you, but they might have. So I think it's that kind of pressure and expectation it is my hope that eventually even the human recording field will release its data. And I think that's necessary and it should be done, because otherwise we will just record the same thing over and over, and that's just a pity. And one other thing that I might want to add to that is that increasingly now in neuroscience, what's happening is that we focus greatly on getting as much data as possible on a very small number of subjects. And we record simultaneously, and then we want to squeeze out as much as we can out of that data set. And under that model, it really makes sense to share the data, because then the majority of the work is really not the experiment. So I think open science is what should be done for that model.
Well, I think that's a perfect way to close this session. So please join me in thanking our panel for their insights.